In the next lecture, we're going to go into detail about the electron transport chain and cellular respiration and other ways in which a cell can create energy storage molecules that can be used to either build or break things within the cell. So, the major players in this whole process and in a lot of cell functions are enzymes. So, I'm just going to introduce you to the structure of enzymes and some basic background information about enzymes so we can move forward in the next lecture with talking more in detail about um, the other processes. So, in general, the idea of metabolism is any type of chemical or physical working that's going on within the cell. We use the term anabolism when we're talking about biosynthesis or building of molecules, and this requires some sort of input of energy. Catabolism is the opposite. So, catabolism is when we're actually breaking bonds and breaking things down from larger molecules to smaller molecules, and this is the release of energy. And in every stage that we go through, either catabolism or anabolism, these processes are conducted by enzymes and enzymatic pathways. So, some of the properties of an enzyme, and I'll kind of just go through these briefly, is that they're made up of proteins and they may require cofactors. And we'll discuss in a second the structure of a protein and how that develops into the structure of an enzyme. They act as a catalyst, so they speed up reactions or they reduce the amount of energy required in order to allow a reaction to move forward. So that's the when you hear it, lower the activation energy. And I'll talk a little bit about what that means in terms of building and breaking bonds in class and how that's so. What's really important to them about is their shape, and their shape is what determines their specificity and whether or not they can function. They enable metabolic reactions. So, a metabolic pathway is a series of enzymes that are required in order to undergo a certain process. And there's all sorts of different pathways. So, it's not just usually one enzyme that's involved in a reaction or the building and breaking of a complex molecule, it's several. They have an active site that is highly specific to a certain substrate, and they're typically a lot larger in size than the substrate. They are sort of an instigator or helper in the reaction, but they're not a part of the reaction. So they aren't used up in the reaction and they don't become a part of the new product. Um, they're not permanently changed during the reaction, so they can be used over and over and over in whatever concentration. They can be affected whether or not they can work based on temperature and pH and some other factors. And they also can be regulated by feedback mechanisms and by genetic mechanisms. And again, I'll go more into detail about a lot of these, but in this one, in this video, I'm just going to talk about the basic structure. So if we go back to the lecture, we discussed chemistry and we discussed macromolecules. We have the hierarchy or the structure of a protein. So it starts with a primary structure where we have peptide bonds linking amino acids. And then we get to the secondary structure where we can see the shape of forming of the proteins. So we start with either an alpha helix as seen here, or sometimes we have a beta pleated sheet, and that's the secondary structure. The tertiary structure is when those alpha helices you see here or beta pleated sheets start to fold over one another. And as they fold over one another, they create these sort of pits and grooves that are active sites. That's what the AS means. And this is the tertiary structure of a protein. And then finally, they come together to form a quaternary or a complex protein structure. So you have several tertiary proteins that come together to form a quaternary structure. And this is really a full shaped enzyme with several different active sites in which there is a very specific shape for a substrate. So we say that an enzyme has a lock and key fit. So based on that really complex quaternary structure, we create an active site that can only fit one specific type of substrate. So you can see here this substrate, which is what the enzyme works on, would fit into this active site, whereas this substrate would not, right? And when the enzyme then is connected to the substrate, then we can either, this would be an example of catabolism, or breaking something apart, or we can put two pieces together. Now, another part of a protein or an enzyme that's required for its function is quite often a metallic cofactor or a coenzyme, and sometimes both. So the apoenzyme is sort of the raw quaternary structure, but then as a regulatory mechanism, sometimes metallic cofactors or coenzymes are required. And this, they really, when they bind to the enzyme, change its shape a bit in order to make the active site more accessible or be the correct shape for the substrate. And so without them, they wouldn't work properly. Keep in mind that really all of this is like an electrochemical interaction. So when a compound binds to an enzyme or to a protein or really anything, it's going to change its shape such that it's causing all of the electrons that are in the large molecule to spread out from one another as far as possible. So by attaching something, you're effectively changing the shape and changing the shape of the active site. So without the metallic cofactor, it would not be the correct shape, or without the coenzyme, it would not be the correct shape. So just kind of give you an idea of the difference between the two. Just in general, metallic cofactors are metals that activate enzymes. Common ones are iron, copper, magnesium, manganese, zinc, cobalt, and selenium. Again, remember we talked about iron before and those magnetotactic bacteria. That iron is a very common cofactor. It helps bring the active site and the substrate closer together. Really, it helps to change the shape of the enzyme, and it participates in the chemical reaction with the enzyme substrate complex. On the other hand, the coenzyme is a larger, sort of like a mini enzyme. It's a larger organic compound that helps to work with the enzyme to sometimes alter the substrate to make it ready for the reaction. Sometimes it will remove a chemical group from the substrate, so it's actually actively involved in what's going on with the changing of the substrate. Um, Vitamins, you always wonder, why do you need your vitamins? Well, vitamins are a component or important component in coenzymes. All right, so hopefully this just gives you a little bit of background information about enzymes so that we can move forward and talk about metabolic pathways and specific metabolic pathways that are used by prokaryotes in order to make ATP.